Your mic.
Hi, everyone. Welcome to our last session for the day. We're going to be closing out today with a session on sex hormones and glaucoma. Our first speaker is Dr. Thasarat Vajaranant, who is a professor of ophthalmology and vice chair for strategic initiatives, director of glaucoma and anterior segment service, and director of data sourcing and strategy at Illinois Eye and Ear Infirmary. She'll be speaking today about epidemiologic studies in support of a relationship between sex hormones and glaucoma.
Thank you very much, and I'd like to thank the organizer and also Dr. Luis Pasquale for the opportunity to be included here, and it's very exciting to hear all these new research and ideas. So this is especially a topic that's a passion of mine, the role of sex hormones in pathogenesis of open-angle glaucoma. So this phase needs no introduction, but did you know that Ms. Jolie may have increased risk for glaucoma? In this talk, we will not focus on Ms. Jolie, but we will focus on what might be, including in sex-specific risk factors for glaucoma. So I do not have any financial conflict of interest related to this talk. Most of the past support that I have support many of the information that I will be sharing today. So in this talk, we will talk about the rationale why we think that the role of sex hormone will be important, and review the epidemiological evidence, and then touch on the impacts and some of the proposed future directions. For the rationales, actually thinking about two, as we know that estrogen receptors present throughout the body, in the brain, in the cardiovascular system, vascular system, breast, and GI-GU system, are there any estrogen receptors in the eyes? So the answer is yes. So there's many studies that show that estrogen receptor present in the ocular tissue, interestingly, both at the site of aqueous production and also site of glaucoma injury. So if we look at the very important risk factor for glaucoma, there's big two. One is age, and the other is intraocular pressure. So for the age, in women, we should not only think about the chronological age, but reproductive aging can also play a role, because it could affect the general health, because the ovarian hormones such as estrogen can also regulate downstream of the organ systems. Secondly, the intraocular pressure, as we just showed, the slide, that estrogen receptors actually present in the ciliary body epithelium and also ocular tissue, and estrogen could also include in the intraocular pressure regulation. So the basic thing first is that any basic science research that support this claim, if you look at these two studies, very elegant study looking at the estrogen that could prevent ganglion cell loss, and the other talk probably will talk more about this, but I just want to show you that both of the animal models that show in chronic IOP elevation and acute IOP elevation also shown that estrogen prevent ganglion cell loss. In these animal models, usually they have the animals that have both of the ovary removed, and then one with the control treated with vehicle, and the other one will be estrogen. You can see that there are preservations of the ganglion cells in the animals that treated with estrogen after ovary has been removed. So now we know that the basic science research does support these roles. How about in epidemiological studies and some of the clinical studies? We're going to look into the association between reproductive factors and the risk for glaucoma, and also whether or not the hormone therapy change that risk and how it's regulated intraocular pressure. Then talk about some of the why that might be the case. So women go through phases during the reproductive phase, and marking at the beginning of the reproduction is the menarche, which is typically occur between age of 10 to 16, on average age of 12. And then end at the age of menopause, in which usually have a transition starting between 45 and then end about 55. The average age of menopause is 51. Having menopause meaning that women have to have 1 year, the whole entire year of not having a period. So there's early epidemiological evidence showing the relationship between reproductive aging or showing that in Rotterdam study is the first study that looking at this and show the early menopause. If women enter menopause before age of 45 have increased risk of glaucoma, and that's the odds ratio of 2.6. Later study, Blue Mountain study, and also a larger cohort study, nursing health study, also supporting that late menarche or also increased risk, or late menopause also decreased risk. So in our work, we leveraged the two databases within the WHI Women Health Initiative, which have a large randomized trial for the hormone trials, and also the Rochester Epidemiology project. This is the funding sources, and I also would like to thank all the collaborators and also mentors, one including Dr. Pasquale. So if you look at this, if we cut down the hormones and women might have increased risk for glaucoma, how about if we replace the hormone by hormone replacement therapy, could that actually reverse it or decrease risk? So we can look at these two different things. So in our study, we have two goals. The first one is to avoid the recall bias. As you know, women might not actually remember when actually they're starting their period or their period stop. So by using the database, such as in the Mayo Clinic cohort study of oophorectomies, we know exactly a time of injury, right? The time that estrogen was taken away. And the second study, or second goal, we also looking at the hormone therapies and to avoid the selection bias or the healthy user bias in the observational studies, and to also minimize the confounding, we conducted the secondary data analysis of the randomized trial of the hormone trials using the data in the WHI. So the first study, we look at the Mayo Clinic cohort study of oophorectomies. This look at the risk of glaucoma after early bilateral oophorectomies. In this study is in the Mayo Clinic cohort, we comparing the women who underwent bilateral oophorectomy from the 1950s to 1987, and compare that with 8 matched reference women who did not undergo either unilateral or bilateral oophorectomies. And then we follow them over time, and in this study is 25 years of the follow-up time, following them up with diagnostic codes of glaucoma through the Rochester Epidemiology project with the record linkage system. So in this study, women who underwent bilateral oophorectomy show no risk for glaucoma. But if you stratify it by age of oophorectomy or the surgical menopause, the women who enter the menopause early before age of 43 have an increased risk for glaucoma, and that's 1.6 times. Approximately 11% of these women had undergone bilateral oophorectomy, also had estrogens up to age 50. In this particular cohort, the study, the analysis didn't find that the treatment reduced the association. So now bringing into how the hormone therapies actually, could it reverse the glaucoma risk? So the earlier studies, the Rotterdam study and Blue Mountain study, which is the cross-sectional study, a population-based study, did not find that risk reduction, but did show some trend. Not until the larger cohort studies, such as Nurses Health Study and also Claim Database, show that using the hormone therapy could actually potentially reduce the risk for glaucoma. So brought us to the second study, looking using the randomized trial of the hormone trial in the Women's Health Initiative. So in our group, we use the database from the WHI, which is a large cohort, have 2 arms. One is observational study, the other is a clinical trial, including hormone trial and then a diet modification intervention. We're looking at some of the association between the optic nerve cupping and structure and functions of the brain. And also the other arm, we're looking at the hormone influence and diet modification from the hormone trial. So at the beginning of the trial, WHI enrollment, after that, 4 years later, there's an ancillary study that looking at the age-related macular degeneration. So we have the photograph and also optic nerve photos, and also complete eye examination, including eye pressure. And then throughout these WHI, we also have the Medicare linkage to follow up for the long term for the clinical outcome. So is estrogen protective against glaucoma? The first one, the study, we look at how does that affect the IOP. The estrogen therapies alone had minimal effect on IOP lowering. That's the first one that we found. The second finding that we found is that estrogen therapy alone reduced the risk for glaucoma, but interestingly, it's only in high-risk women, and that is in African Americans. So now we're gonna look into how we did that one. So the first one is the hormone trial of the WISE, which is the WHI site examination, included 4,347 participants and have two different arms. The first arm is that if the women have no uterus or already have a hysterectomy, that group of patients can have estrogen alone. But if they still have uterus, they need progestin to counteract the estrogen to prevent the endometrial cancer, and that's the common practice then. And then we randomize patient into the one with treatment and the other with placebo. In these multivariate analyses, we are comparing to placebo, the treatment of estrogen alone associated with 0.5 millimole mercury reduction of the IOP, and that's similarly found also in the right eye and the left eye. Interestingly, in the other arm with estrogen and progestin trial, we did not find any significant differences. So how is that translate into, would that also protect the patient from having glaucoma long-term? Because this we can also not only during the intervention period, which is 4 years, but we can follow up 25 years later with, not 25, this one is 12 years later, whether or not the patient will develop glaucoma. So in this trial, we look at the WHI trial with the hormone trial starting with 27,000 patients, with the look back and also excluding some of the prevalence, also we ended up with final analysis of about 8,000 women. And then we look at the incident open-angle glaucoma by the ICD code. In the second data analysis, we found that the risk is reduced, actually varies by race. Overall model, we did not find that the hormone therapy reduced the risk of glaucoma, but when we stratified by race, we found that among African American women, this risk is reduced by half, or the hazard ratio of 0.49. So wouldn't it be nice if the patient have risk reduction cut by half, right? So this is the forest plot that's showing that only the African American women that would benefit from the hormone therapy in this cohort, and that's with the follow-up time of 12 years. So now this is a recent systematic review in ophthalmology glaucoma last year. I think this is an excellent review looking into how the female reproductive factors associated with risk for glaucoma and also related traits such as IOP. So of all the literature search, they included 27 articles in this study. They also performed the risk of bias assessment, looking at the different types of study that included trying to answer two questions. One is association between female reproductive factors in glaucoma, and the other looking at the association between the hormone therapy and how is that affect the intraocular pressure. As you can see, these different type of designs on this study, the green dots represent less bias. So as you can imagine, the randomized trial would have less bias in cohort study compared to the cross-sectional study. For the IOP, the control study, which have a control arm, would have less bias compared to the study that have uncontrolled arm. So now we're going to look at the association between the female reproductive factors and open-angle glaucoma. So starting with Menard, there's 5 studies that look into this. The evidence suggests no clear association, but the NurseCell study is the one that showed that late Menard was associated with a slightly higher risk for normal tension glaucoma. For the menopause, There's more study and including into this is 7 study. Overall, the direction and consistency findings suggest of the association between the younger age of menopause and also higher risk for glaucoma. If you think about the total entirety of the reproductive phase, that beginning with the menarche and end with the menopause, that is the time that patients would expose to the estrogen. So these could be the surrogate of the estrogen exposure. So there's a study, 4 study that look at the years of reproductive duration and look at that risk for glaucoma, and the association remain unclear. Only 2 large study that, only one Vajaranant study that show that they suggest that additional age of reproductive duration associated with 5% reduction of the odds of the open-angle glaucoma. But NurseCell study, which is the largest cohort, did not support this. And if you look at during the time that the women during the reproductive phase, some women would elect to have the oral contraception and also having babies or pregnancy, and that actually also interrupt with the normal hormones balance as well. And many studies have looked into that association. So for oral contraception use, the total study of 4, the existing literature does not support the direct association, but there's also some trend of the longer duration of OC use, increased risk for glaucoma. How about pregnancies, the number of pregnancy? There's 5 papers that look into this. In the 2 study that show the associations are Blue Mountains Eye Studies and Korean National Health Nutrition Examination Survey show some association. But the conclusion was that no likely direct relationship because of the doing these. Here is the many ways that we can use as a surrogate of the estrogen exposures, and the one that particularly study and the one that have more consistent data that show association is the age at menopause. The lower or younger age in menopause increased risk for glaucoma. So then it's not finished yet, right? So after menopause, some women can also elect to have the hormone therapy, and that hormone therapy can also influence the hormones after the menopause. Could that actually be a protective or delay the time that the patient can have glaucoma? So there are many studies that looking into this, including 11 studies, different type of study, randomized clinical trials, cohort study, population-based study. In general, population-based study did not show the association, but the large cohort studies such as the NurseCell study or the WHI, the randomized trial study, show that the hormone therapy could be protective. And that actually, interestingly, it depends on the type of the hormone therapy. And being said, estrogen only seem to have the effect of the neuroprotection, and the race also, depending on the race, and like in WHI found that among African Americans, the effects seem to be more significant. So looking into the IOP and relationship with the hormone therapies, there's 15 studies that looking into this, including control study, uncontrolled study, and some study have pre and post pressure once you start a hormone therapy. This is another one that most consistently found that most suggests that association between the use of hormone therapy and the lower mean of IOP. And the type seem to be consistently showing that estrogen alone seem to have the best effect. In our study, we found 0.5 millimole mercury and noted that that particular group of women also much older. In other study, the ranges of IOP effect is around 0.1 to 3.8 millimeter mercury reduction. So now we have both basic science research to support this, and also many good study of the epidemiological study to support the role of female sex hormones. So we might think why that might be the case. We propose the theory of estrogen deficiency, just like any other cell types in the body, accelerated the aging and also accelerated the aging of the optic nerve. Therefore, it's increased risk for glaucoma. So I'm actually was excited when I saw Dr. Weinreb's slide because this is one of his slides as well. So I call that a success for today. This is including the pathogenesis of glaucoma with different, theory that included, and that boiled down to this diagram with the mechanical theory, vascular theory, and some abnormal immunities and glares or some inflammatory component. And if you think about how aging have such a big impact in increased risk for glaucoma, if you're looking by mechanism, the mechanical theory, you might have this less flexibility at the lamina cribrosa. For the vascular theory, you might have more compromised blood flow. And for the reserve of the retinal nerve fibrillary or ganglion cells, we know that as we age, these cells die. So we have less or lower in our bank. So whatever injury that we might have, that might push us over the edge and might be the risk for developing glaucoma. And also lastly is the support system is less, so will be less of the to fight the oxidative stress. So that's how the aging could increase risk of glaucoma by many different mechanisms. So now we're going to look at why estrogen deficiency or hormone therapy could that relate to accelerating aging, and how could that hormone therapy could protect it. So you look at the mechanical theory, estrogen may regulate IOP. We have that evidence also shown in many studies showing the higher IOP in postmenopausal women, and also many studies that I just presented suggesting that IOP reduction can be found after the hormone therapy. How about the vascular theories? The estrogen also regulates smooth muscle tones and through the endothelial-based nitric oxide synthase, and that might also influence the perfusion. The aging and age-related decreases in female sex hormones also could decrease the blood flow. And another study also found that hormone therapy also could enhance the ocular blood flow. And Dr. Harris worked with your study as well. How about the underlying, right? Aging, could lack of hormone make this worse? Our study looking at the women who have early oophorectomy show that they do have thinner retinal nerve fibrillary compared to the women who did not have oophorectomy at younger age. And hormone therapy in another study actually reversed that, or could preserve the retinal nerve fibrillary. So we have these multiple sources of evidence showing that estrogen deficiency could perhaps accelerate the aging of the optic nerve, and that increased aging, meaning it's increased the risk for glaucoma. And some of the study also support that the hormone therapy could reduce that risk. It will be interesting also to look at the effect of estrogen on the lamina cribrosa or other tissue as well. So now we have all these evidence from a different type of study. So how about the impact? Would that matter? Why women? So we looking at the prediction and also the current cases of glaucoma. We know that glaucoma is more common among minorities, African Americans and Hispanics, but if you multiply that by a prevalence rate, the majority of patients who actually having glaucoma is non-Hispanic white women. So that is the huge caseload. And also we know that women have a higher rate of visual impairment, and also there's some disparities in treatment, and that will be even more enhanced in the minorities women. So how big is this? What the early menopause, the people who we're talking about might have increased risk for glaucoma. The 6% of women enter menopause before age of 45, and in 2010, we estimated that would be 10 million women that perhaps have additional risk for glaucoma. And not only that, in addition to that risk, the higher risk of premature menopause also occur in women who underwent hysterectomy or uterine artery embolization at younger age. And the thought is that maybe that surgery or that particular the intervention is interfere with the vasculature of the ovaries. Lastly, the medical conditions such as cancer that the patient require to use some anti-estrogen treatment for long term, could that be also predispose them to have early aging of the optic nerve, therefore might increase their risk also for glaucoma. So now we're coming back to Miss Jolie. So now you probably can tell that she anyone know her stories about why that? Yes. Yeah, she went quite public about her decision to prevent ovarian cancer because that's run in her family, and her mother died from ovarian cancer. She is BRCA gene positive, so she elected to have bilateral oophorectomy done at age of 39. So in some of these moments, they should also know additional risk when they decide to prevent, one disease, and then you might run into getting increased risk for different conditions. So how about what our colleagues said? This is from the NAM's position statement from North American Menopause Society last year, looking at the hormone therapy in terms of recommendation. So they're actually looking at our study. So we need more study to kind of also guide them as well. So they quote the study from the WHI as well, with the estrogen therapy appear to reduce intraocular pressure and also can reduce the risk for glaucoma, and especially the effect is higher in Black women. But should we then recommend everyone to have hormone therapy? It's not as simple, because risk for hormone therapy also differ for women depending on different things: type, dosage, duration of use, route of administration. And I think the most important thing, the one that I highlighted in red, is the timing of initiation. And that's called timing hypothesis. And many studies in different fields consistently showing that if you give the women at younger age, that effects seem to be positive. But if you give that later, 10 years, 20 years after, or when they actually older at age of 60, that risk benefit ratio seem to be less favorable. And that I thought, when I listened to the neuroprotection talk, maybe it's just the time of injury, right? Like you have to rescue it. And this might be somewhat a very similar situation. So in closing, women bear a greater risk for glaucoma by, have more of a caseload, and also higher rate of visual impairment in general, disparity in treatment. We have the mounting evidence of the role of sex hormone in pathophysiology of glaucoma, and looking more into this might also improve our care for glaucoma, and could serve as a foundation for personalized glaucoma care, develop the new treatment, and also have insight into the therapeutic target that could benefit men as well, hopefully in the future. For the future direction, we could also explore this in different patient population with diverse, also in men, looking at the hormonal influence, and also including more genetic epidemiology study to look into more insight into race and also hormone therapy and IOP relationship. Thank you very much.
Okay, so our next speaker is going to be Yutao Liu, who is an associate professor and the director of cellular biology and anatomy at Center for Biotechnology at the Medical College of Georgia. He'll be speaking to us today about estrogen signaling pathways in glaucoma.
Good afternoon, everyone. It's a really great honor here to share with you about my story, I think 4 years ago. So it actually comes with, through a different path. Yes, so this is, if you have ever played golf, as you probably know this place in Augusta. So actually, I didn't know this city until I moved there in 2014. So this is near the Savannah River. We have a satellite campus for cybersecurity, and there's cyber commands from the military. This is new medical school building. So if you don't know the Medical College Georgia, because Augusta University is really new. We changed the name 3 times in the last 10 years. So I think this slide is everyone knows. This is, so by training, I am a human geneticist doing GWAS linkage, sequencing, expression, omics. So how I came to estrogen pathway is really the story through this. So we all know about this, aqueous humor homeostasis, through the conventional and unconventional pathway, mostly through the TM and Schlemm's canal. And so this is the current date of the NGRI, European Bioinformatics Institute. The GWAS catalog, almost every chromosome, every loci has these total things. But back in 2017, through the neighborhood, and so with my old mentor Rand, so we did a experimental research review in collaboration with the CC Core in Singapore. This is in 2017. There are only a handful of genes, but then in 2021, so we have 127 POG loci, and the story just showed, now we have over 350. So it's really overwhelming. In the early days, people in the neighborhood, people said, "Hey, this is my gene, that is my gene." So it's really, so we try to coordinate it, but now we have so many genes to work on. So POG is a really complex disease. We try to find diagnosis early on. So we still have different opinions after 20-some years. At least IOP is not part of the definition, but it's still a significant, only modifiable risk factor. So we start with these two, large genome studies back in 2018. And so this is the work from Anthony. And so with 112 genomic loci, 68 of them are novel. And then Stewart also found, 101 genomic loci. So really, what can we do? Also, we have a new NER director, Michael Chang. So from, they always follow up with the new, strategic plans. And this is really guide us, me at least, how to really look at my next steps in the next 10 years. And so this is, many people here are actually involved in the consortium to define these strategic goals. One is the top one. Number one is from genes to disease mechanism. So imagine with all the consortium, NEI put hundreds of millions of dollars over the last 20 years into the genetics with all the genes. They definitely want to know what those genes do. And so I think this try to, align my research to this strategic plan. So I have a really young and star student, Hannah Youngblood, who is doing a postdoc with Ross Ether at Georgia Tech right now. And so basically, this is back in 2010. We designed a kind of a prioritization plan to really look at these, 157 IOP-associated genes from the two studies. And we look at through all the, our own and public available expression data sets. And then also we did a correlation between glaucoma genes and also the IOP associated genes. With that, we did a number of different bioinformatics analysis, and we narrowed down to a few pathways surprisingly. So the genes highlighted in black here are the IOP associated genes. And then the center you can see is ESR1, that's also the estrogen receptor alpha. And then also dexamethasone, that's a high risk factor for glaucoma. So this is really indicating something. And also estrogen receptor in general, also targeting many genes in the IOP associated genes here. So this is really, we see another one, ESR1. So this is really opens, okay, what's going on there? And then we look at the estradiol presence in the aqueous humor from bone eyes, from porcine eyes, and also from glaucoma, non-glaucoma aqueous humors. And we do find really the presence of estradiol, the active form, the E2. And then we look at the expression of ESR1, so also present in the aqueous humor pathway there, so outflow, the TM and Schlemm canal. And we also see similar things in the mouse tissue. And then also, the eye is really under constant ocular pulse. It's a kind of enclosed ball structure. Every time we have the pulse, so it's kind of pumps in that way. We call it ocular pulse. So through collaboration with Dan, so we did actually, cyclic mechanical stretching of TM cells using the flow, the flex cell station. And so basically you have this TM cell put on the membrane and get stretched every second. And so by 15%. And so we do that for 24 hours, and then we collect the cells. And so we look at their expression. We did the RNA sequencing, and we found actually, the steroid biosynthesis pathway keep popping up, and also lipid metabolism. And so actually now really produce work from Indiana University also support this. And so we look at more, we saw steroid biosynthesis pathway. And so then we see also, another receptor called the G protein coupled estrogen receptor 1. So this is previously thought is kind of a really a receptor called GPR30. And so we see significant there. So kind of at this stage, okay, our data suggests this involvement. So what are they? So Thasarat already mentioned the two receptors, alpha and beta. They are nuclear receptors. They really present in the cytosol and cell membrane, and it's functioning through genomic and non-genomic response. And also the G protein coupled receptor is really kind of novel. It's a 7 transmembrane protein located on the cell membrane and ER membrane. So it's really, and this is just, if you pay attention just to the ER alpha and the beta, so they are nuclear receptor, and they so really, here they have different isoforms. Alpha has 3 different isoforms, beta have 5 different isoforms, and then GPR1 has 1 isoform. This is probably, I try to use this, maybe explain the things that we observe in human. And also the synthesis of estrogen. So basically this is very simplified one. And so DHEA, many people study, and then also estrogen, the estrogen, and then that's the E3 and the testosterone. So testosterone present in both men and women in high levels in the serum. And so only convert differently, either for estrogen receptor or androgen receptor. So in this way, it's really hard to distinguish really either men or women receptor is really, and this really kind of shows you more. So with estrogen, this is synthesis. You see, yes, estrogen in the plasma is mostly in women, is premenopause. And then after that, so definitely you see it's very different. And so this is all increase the complexity. And this is kind of, we talk about 2 level, 2 different words, ligands versus receptor. And here just shows, really you have just 3 receptors here, but look at all these things here. One thing, everyone try to avoid is the BPA, so DTT, DDT, so tamoxifen, how many people use it for inducible Cray mice in their studies. So really kind of, eye-opening for me to learn all this. So here shows you the 3 different isoforms for the alpha receptor and the beta receptor. Look at their difference is based on the ligand binding domain on the C-terminal. So these different isoforms actually determines a ligand whether it's agonist or antagonist. So this is really at a different stage, I assume with different people may have different effects in this way. And then this is just ER alpha shows, you have the regular genomic pathway. So go to the estradiol, go to the cell cytosol, bind to the receptor, and then go to the translate to the nucleus, bind to the DNA, and then turn on the transcription, and all bind to the other transcription factors, and then regulate the downstream regulation. Or, this is kind of ER receptor also on the membrane, kind of go through these secondary messengers, or they could go through other, non-receptor dependent. And so, so this is really make it more complicated. And this is G protein coupled estrogen receptor 1. So this is really the receptor is on the membrane, and so works through the G protein. So it happens within 3 minutes or 30 seconds, really quick through the kinase pathway. It's a really, so in combination, the first one is this really the #1 genomic response in the cytosol, and then you could have estrogen receptor bind to the transcription factors to the DNA. That's the genomic still. And then you could have the cellular membrane receptors bind to these and through the kinase pathway here. And then also you could have the 4th one, the growth hormone receptors, and then through, indirectly activate the estrogen receptor. So it's really kind of made my brain exploding. So it's really, okay, what's to do? So we look at the estrogen receptors in the TM cells from the primary, the coronary rims tissues. And we look at 3 male, 3 female, so yes, very small number compared to beta-actin. So these are beta-actin levels, so relative to that. So we see definitely ESR1 is more stable in the female compared to male, and GPR1 is really kind of consistent between male and females. And then we look at the, these are primary TM cells versus Schlemm canal endothelial cells from Dan's lab. So we do see ESR1, GPR1 more present in the cells. And then we also look at, from the glaucoma, the ocular tissue bank database online from Iowa, and also from Mike Hauser's previous study during RNSeq from the TM and cornea, we do see ESR1 and GPR1 are consistently highly expressed. Then we look at the stretching response. We do see GPR1 is more significantly responsive to that. And then we also look at the TM tissue back in our 2013 study. So we do see they are present. GPR1 is much higher. Same thing with the Schlemm canal endothelial cells. And also with Wanzio's single cell study, we did look at this. And so the red arrow indicates that this is human and this is mouse. And we do see ESR1 and GPR1 are really present in the TM and Schlemm canal cells there. And so this is really says, okay, we're in the right direction. We're not really off track. And then we designed a kind of a cell culture study. And so we take 10 primary TM cells from 10 different donors. So in this case, 6 females, 4 males, and then we put them on those stretching plate. So we have, each plate, we have a medium control, and then DMSO vehicle control, TGF-beta2 estradiol, and TGF-beta2 plus estradiol. And so everything goes through this, and so we have a total of 100 samples. And so we did RNSeq. And so this is the experiment design using RNSeq. And so interestingly, when we compare the estradiol alone with control, medium control, there are almost no gene undergoing any differential expression changes. And so alone doesn't really do much. And so here, basically, this is just one of the conditions if we look at all 10 cells under stretch or non-stretch. And so you see this, here, TGF-beta2, the orange circle here, we have over 300 genes are differentially expressed. And after adding estradiol, and we do see there's only, this number of genes remain significant. All these 285 are not significant. So that's the interpretation of these figures. Same thing here for the static one. So around 300 genes differentially expressed by TGF-beta2, but adding estradiol, and so all these 115 are not significant anymore. So this means there's anti-TGF-beta effect from estradiol. I mean, this is not new. Cancer research already proved this. And so we just repeating the same things in the TM cells, either in the female or in the male. And so we say less in the male, probably due to power issue, because sometimes with 4 and 6, there could be a significant power difference. And we also, under stretch, we do see, and also we found actually stretch induce the TGF-beta response. So this is showing the same thing. So male, so we still see much less impact here versus, so we see, we still see half of the genes here, and then in female, we see 300 genes, so dramatically. So when we do the pathway analysis, and we do see, pathways highlight up with the cell adhesion molecules, and then also acting cytoskeleton. And also, not surprisingly, we do see TGF-beta pathway popped up, and also estrogen receptor. And so these are related to cell stiffness, stretch fiber formation, and also response to IOP changes in the stretching condition. So with the stretched one, so we do see steroid hormone biosynthesis and also P450 activity here. So not surprising, we see estrogen receptor and collagens are really highlighted here. So then also we see protective against the glaucoma process here, and then crosstalk with TGF-beta pathway. PR3K, this is really downstream of the estrogen receptor. And so we also see the SMAD, and so this is based on the RNA data. And so then also recently from Simon Herberg shows the YAP and TAS also involved in the response of TM cells, and we do see here could be a potential regulator by TGF-beta 2. And so this is just show you, give you a few samples here. So we do see here with the TGF-beta treatment, we do see significant change for this gene, TGF-beta receptor 1, then adding the estradiol really reverse this change to non-significance. So same thing for GPR1, so really brought the receptor expression up significantly and become non-significant compared to the controls. And then the one of the SLC711, this is one of the TGF-beta response genes. And then also INHBA, also involved in TGF-beta pathway. And so in the stretch condition, we do see, and so really correct this for the GPR1. And so same thing showing here. And okay, so basically we do see this anti-TGF-beta activity by the estradiol treatment at this concentration. So this is really kind of give us, okay, we are still moving forward. And so come back here with this pathway, we have this estrogen biosynthesis. And so the aromatase is really the main enzyme here. So actually many years ago, Lou and his colleague at Harvard actually already did this by knocking out the aromatase in the mice. And so in the females at 12 and 24 weeks, they do see elevated IOP by 2 millimeters mercury. And so they were able to see reduced RDC counts. And so at early stage. So how about the ESR1 mice? And so we get these are global knockout mice, and so we get from Jackson Lab. And so we age them, and we start IOP measurement weekly from 3 months. We follow them weekly. And so it's definitely a lot of work to 12 months. So start from 8 months and 9 months, 10, 11 months, we do see IOP increase about 1 to 1.5 millimeter mercury increase. So this seems like only present in female and homozygous. And the homozygous mice are super tiny, and so we can tell by the phenotype. How about the GPR1 knockout mice, the GPR30? So we get mice, these are global knockout, they are normal, they're okay. And so we measure their IOP start from 3 months to about 10 months. So this is just male, female combined. So we have about 40 mice in this way. So we try to do 15 to 20 mice per sex, per genotype, after our discussion with Simon John. And so we do see, so this is all the ages, from 3 months to 10 months, we do see wild type, and then the heterozygous, homozygous, then we combine every animal as one dot here, combine all the times together. So we do see there's no difference between the heterozygous, homozygous. This suggests a dominant model and also not sex specific or not. So if we look at males and we see similar pattern. So here, if we look at females, yes, it's still the significant elevation here. So it's really, we see GPR1 shows a non-sex specific and also dominant pattern, compared to the recessive one in the ESR1 knockout. So in this way, so I think there's some animal data to support this, and so there's more work need to be done. So really, we just, I think from a different point of view to show estrogen signal is significant, but it's really complicated with the ligands and with the receptors and different isoforms. We don't even know what isoforms are present in the human TM cells, I think. So definitely we are doing more with in vitro and in vivo, try to use different agonists, antagonists to figure out what's downstream part. So per suggestion with Lou, we are really think estradiol actually upstream of the enos, and so we have interesting data to show actually there's interaction with the GPR1 and then NO pathway. And so we also try to do all the, the downstream omics study. So with that, I cannot be here with all the work from my current lab and then also the former labs and also all the wonderful collaborators and the fantastic supporters, including Glaucoma Foundation here, supporting me 3 times from 2010. So thank you very much.
Okay, our last speaker is Andrew Feola, who is an assistant professor at the Emory University and a research biologist at Atlanta VA Medical Center. He's going to discuss how hormonal changes may affect the development and progression of glaucoma from a biomechanical standpoint.
Oops, thank you very much for that introduction, and thank you for the Glaucoma Foundation for the invitation to come here, and Lou for actually giving some attention, obviously, to women's health issues in this area, because he's been doing research in that for a long time. I would like to point out that while I claim biomechanics is the argument, this is kind of a gateway drug for me because I'm actually a bioengineer. So that's where it started out. So we'll just talk a little bit about biomechanics, and we're going to go into a very different direction very, very quickly. And I'd like to thank, obviously, the presentations before me. They set up everything from an epidemiological clinical standpoint to an estrogen signaling standpoint as well. So I don't need to go into this a lot at all because it's been summarized. The timing of menopause is related to developing glaucoma. Estrogen receptor beta mutations are associated with ocular hypertension, and specifically glaucoma, open-angle glaucoma in females, and that polymorphisms in estrogen metabolic pathway are also associated with the development of glaucoma. So as summarized before, menopause is kind of in a very general sense defined as 12 months without menstruation and occurs on average at the age of 51 years old. Each day in the United States, 6,000 women become postmenopausal. So that's over 2 million women a year that become that in the United States, more if it's a leap year, obviously. And if you think about it, we know menopause affects systems throughout the entire body. It affects cognitive function, hair loss, thermoregulation, cardiovascular health, pelvic floor health, bone, cartilage. So it's odd to me that we think it literally affects the hair on our heads to the toes, like the nails on our toes, and it would skip over the eye. That seems highly unlikely to me. So I think we need to assess if menopause is associated with factors related to the development of glaucoma. So why is this a women's health issue? So if you summarize all glaucoma subtypes and everything, a majority of those cases are female, 59% of the population. And as far as I know, while aging is important, 100% of aging women will become menopausal if they live long enough in their lifetime. Furthermore, as highlighted in the earlier talk, early menopause is associated with a higher risk of developing glaucoma, and that affects 6% of women. So it's very important, I think, to understand something that affects 100% of the majority of the glaucomatous population. Any understanding in that would probably help our understanding of glaucoma overall. So we're going to talk about, again, first the biomechanics, because this is how I got started in our research and where my fundamental interests lie. So the fundamental question we ask is, does surgical menopause impact ocular mechanics? And those of you who don't know me, I actually started out in urogynecology, so a very, very different field than ocular health. So I studied the effect of pregnancy, menopause, and hormone replacement therapy on biomechanics of the pelvic floor. So from that lab, this wasn't my work, but from those studies, they found that surgical menopause in young animals decreased biomechanical properties of the pelvic floor. So here we just have like normalized stiffness on the y-axis, controls that dashed line across. So in surgical menopausal animals, there was a 40% decrease in the stiffness of the whole pelvic floor. But what was interesting is that could be prevented with just giving estrogen or estrogen plus progesterone. So that 100% basically mitigated any significant change after menopause. Follow-up studies found that estrogen altered matrix metalloprotease, or MMP, production and activity. And those of you who are aware, MMPs play a very important role in the extracellular matrix, which then eventually will lead to the biomechanical properties or the functional properties of that tissue. So because that was my background, I started in the eye research, and I basically read some of Lou's papers in the Epidemiological, and I was like, this seems like something that we should probably look at. So I took young, middle-aged female Brunori rats, and they underwent sham surgery, ovarectomy. So those who are unaware, ovarectomy is just the surgical removal of the ovaries at a specific time point. After 8 weeks, we assessed ocular compliance, which is just the inverse of the stiffness. So a higher compliance is actually a less stiff tissue. And we did this using the eye perfusion system developed at Imperial College. So basically what this allows us to do is control intraocular pressure, measure the flow going into the eye while we're simultaneously measuring pressure. So it's also important to note that ocular compliance varies as a function of pressure. So here on the y-axis, we have ocular compliance versus pressure on the x. And you can see that compliance decreases, pressure increases. And this is intuitive when you think about when you're blowing up a balloon, right? Eventually you're blowing up a balloon and you can blow as much as you want. The balloon is really resisting that it's not going to expand anymore. And that's really what's kind of happening here. So to do this, what we did is we actually model it with a function, and we determine ocular compliance at a specific reference pressure. And for this case, we took 20 millimeters of mercury because that's the average intraocular pressure of a brown nori rat. So in summary, really obvious to us was that we found an increase in ocular compliance. So basically there was a 15% increase in ocular compliance after surgical menopause, indicating that they're actually, remember this is less stiff tissue all of a sudden, which means it would deform more at any given intraocular pressure. So kind of following this is not temporally how experiments happen, but this is the next logical question. Does surgical menopause then influence gene expression in the sclera? So what we did here is we have young sham animals, young overrectomized animals, and middle-aged animals. We take their animals 8 weeks after surgery again. We isolate the sclera. We did the retina too, so don't mind that bottom thing, but for this experiment, we're going to talk about the sclera. We did bulk RNA sequencing, and then we did differential expression doing gene set variation analysis. And so really the two things we're kind of comparing here are the effect of surgical menopause. So we compared the young overrectomized animals to the young sham animals. And we want to differentially know if there's an effect of aging. So we compared the middle-aged animals and middle-aged sham animals to the young sham animals. So what's very interesting here is that we just look at, we kind of highlighted extracellular matrix genes because we're looking again at scleral biomechanics at the end. So we took extracellular matrix genes, TNF alpha genes, and highlighted in the previous talk, TGF beta genes are highly regulated by estrogen. And we took all those genes, we took the top 20, and we just said, okay, do they actually cluster? Is estrogen, do menopausal animals have kind of this clustering you'd expect if there's actually an effect? So what you see here is there is clustering. Most of the overrectomized animals clustered nicely on one side. There's one animal that really apparently didn't have an effect of overrectomy, as like no genes changed. But I mean, that's biological variability. Like it can happen. Like a person might not be as sensitive to hormones as another. So I take that as proof that we did something right while it came out wrong. So we look at the effect of age as well, and we can actually see again, if you just look at young sham versus aged sham animals, so the aged sham here are in green versus young sham, there's again a good clustering based on genes. So that's interesting. So then we did like gene set of variation just on the extracellular matrix remodeling. We're not going to go all through the gene sets here at all, but I think the important thing to note here is that they had opposite effects. So all the gene sets associated with extracellular matrix remodeling after surgical menopause were upregulated, indicating there's a lot of things going on. Whereas when you compare aged sham versus young sham, all of those genes are now they're not as enriched, so they're not as active as they were once before. And while the age one makes sense because there's less turnover when you age, things get stiffer, we're still trying to figure out what the change in surgical menopause happens, or what that means. So the question then obviously was, does menopause modulate intraocular pressure, which I think has been answered a little bit, but it's important to recall that, as we all are aware, elevated intraocular pressure is a major cause of risk factor for developing glaucoma. This is an age-old study from '91 looking at the relative risk of developing glaucoma versus intraocular pressure. And it's important to point out that menopause and estrogen specifically, it can cause modest changes in intraocular pressure. And we're only talking about 1 to 3 millimeters of mercury change when one becomes postmenopausal. But I think what's important, and I just try to iterate this by bringing this graph up, is because, yeah, that doesn't seem like a lot, but if you had 15 millimeters of mercury IOP and you have this 1 to 3 millimeter mercury change in IOP, that's a relative 2-fold increase in your risk of developing glaucoma. worse if you had a 19 to 20 millimeter of mercury range and you have that modest increase in intraocular pressure, which would be associated on this graph as a 10-fold increase in your relative risk of developing glaucoma. And I highlight this because once you go through menopause, it's not a one and done event. You don't have an elevation in intraocular pressure for a day and then it goes back down. This is now a chronic sustained elevation in intraocular pressure. So if you think elevations in intraocular pressure have a role in developing glaucoma, something that modulates intraocular pressures probably also plays an important role. So then again, we go back to, okay, well, let's tease out this, like what's going on here. So does menopause affect factors important for regulating intraocular pressure? So the major factors for IOP regulation are formation of aqueous humor, as I think we're all aware, aqueous humor outflow facility, and episcleral venous pressure. So this obviously gives full to the Goldman equation. So if you have intraocular pressure, basically would equal, aqueous humor formation divided by outflow facility plus episcleral venous pressure. And so we'll focus on outflow facility, largely because no studies have, at least statistically, shown changes in aqueous humor production in females as they age. And episcleral venous pressure has been shown to be relatively stable over time. So, Yahtzee, let's hope it's outflow facility. As Yutao just summarized, estrogen plays a role in the outflow pathway. So there's estrogen receptors present in the human outflow pathway. And again, estrogen is an upstream regulator of genes associated with regulating IOP. So there seems to be something pointing in this direction on a pretty regular basis. And there was these studies. So if you go back to literature and you try to go back, this is literally before IRBs. So I don't think this study would happen ever again. But they measured IOP and outflow facility in hysterectomized women. And this is when hysterectomized included a free ovarectomy. And then prescribed estrogen or combined with progesterone for 6 months. So they measured outflow facility and IOP. And what they found is that there was a 30% increase in outflow facility and a 2 millimeter mercury decrease in IOP at 6 months. You kind of notice this trend from my talk. This estrogen menopause thing is right in that range of very consistency, 1 to 3 millimeters of mercury. But what's really cool about the Goldman equation, right? And I'm an engineer at heart, so I like to use equations for things. You have an IOP, you have estimates of aqueous humor inflow and episcleral venous pressure. So if I plug in what outflow facility they measured, I should estimate what they measured for IOP, right? That would make sense. So if you take IOP in the premenopausal women, plug in their outflow facility, they get 15 to 21 millimeters mercury. Spot on, fantastic. If I do it for their postmenopausal or their hysterectomized women, they had an IOP post between 13 and 18 millimeters of mercury. That's with treatment, sorry, I guess that's post-treatment, not post-hysterectomy. And that's exactly what they measured. So that makes perfect sense. The percent change in outflow facility matched exactly the change in IOP based on this equation. So then we're like, okay, well, let's go back to the rats because that's what I'm most comfortable with. This is basic science speaking here. So does surgical menopause impact IOP and outflow facility? So the first thing we did is we took surgical menopause animals and we found an increased IOP. So we took these animals, we measured IOP kind of weekly. We had one group that was overrectomized, one group that was sham, and we saw a modest elevation of, again, 2 millimeters of mercury in rats that were overrectomized. So then we looked at outflow facility. And again, outflow facility varies as a function of pressure. And so we have this equation that we fit to it, and then we determine outflow facility at a reference pressure. So this we use in a reference pressure of about 13 millimeters of mercury, or 12 millimeters of mercury, because that's IOP minus episcleral venous pressure. And we found that there was a 34% decrease in outflow facility after surgical menopause in animals. Okay, so if we review a little bit, we found an IOP increase of 2 millimeters of mercury in overrectomized animals. That was similar to clinical studies. Spot on, that's good. We found a 34% decrease in outflow facility after overrectomy, which is similar to the percent change they found when they gave women estrogen. So that's really good. So this is indicating that maybe it's a good model, we'll see. So then we go right back to the Goldman equation. We say, okay, if we know an estimates of aqueous humor inflow and episcleral venous pressure, plug in our values for outflow facility, what do we get? Okay, in the sham animals, it should be about 22 millimeters of mercury. That's exactly what we get. If we look at our overrectomized animals, we do not get 31 millimeters of mercury. So we have absolutely no idea why the Goldman equation would estimate that so differently. So there's possible things that are going on here, right? There's potential compensations for outflow in young and middle-aged animals that we're unaware of. Obviously, there could be differences between menopause versus estrogen treatment that we're just unaware of at this moment as well on both IOP and outflow facility. And this is a figure adopted from Stamer and Ethier from one of their papers, where they have aqueous humor inflow. You have the outflow resistance generating intraocular pressure, which causes stretch and shear stress on the tissues. Then there's obviously a change in signaling in the outflow pathway. And so this could lead to changes in inner wall permeability, extracellular matrix turnover, and contractility. And I just point out again that we did do some RNA-seq on the effect of menopause, and we do see that there's changes in extracellular matrix turnover, at least in the sclera. And the same thing was true in the retina. So I'll extrapolate, and I'm assuming that it will affect the outflow pathway, but we don't know that quite yet. But there are some things going on here that we don't quite understand. So relevance from menopause and glaucoma, so far, basic science studies have actually also shown that menopause affects intraocular pressure, ocular mechanics, outflow resistance or outflow facility, and gene expression. So these are all things that are related probably to development of glaucoma. So then the last thing we're going to talk about here is, does surgical menopause impact visual function after retinal ganglion cell injury? And I think this is important because a lot of the things I've shown you before are basically just looking at the effect of menopause. There's no injury, there's nothing happening. So these are just baseline changes between menopause and age. So what we did is we have two models. We have an ocular hypertensive model where we injected hypertonic saline in episclera venous outflow pathway. This is the Morrison model of ocular hypertension. So that's an IOP dependent model, like you're developing ocular hypertension. So IOP dependent model of retinal ganglion cell loss. And we've actually done it as well in an optic nerve crush model, which is an IOP independent model of retinal ganglion cell injury. So obviously, again, this is in rodents, so you can't ask them to specifically look at a vision chart and say, can you read these letters? So we assess visual function using an optomotor response. So in this, we take a rodent, we place it in the center of the field here. And basically, you have 4 monitors that create a virtual drum around it that have varying degrees of spatial frequency. So that's like here, right? So this would be like the biggest letter. This would be the smallest letter on the visual acuity test. And you basically change the thickness of those bars until they no longer respond. So you're relying on an innate response of an animal that when a signal comes from the peripheral, they'll actually track it. We actually also have that, and obviously. So it's something very ingrained in animals for vision. So you can actually track one eye. What's nice about this is we can induce injury in one eye, and we can assess visual function in one eye. So we have internal controls versus their injured eye. So what we look at here, we have spatial frequency on the y-axis. Higher values are better. So we're looking at ocular hypertension. The dashed lines represent the contralateral eyes. Red is the overrectomized animals, blue are the sham. And what you can kind of see is from baseline through 8 weeks of ocular hypertension, there's no real change in the control eyes, which is good for controls. And then obviously after 4 and 8 weeks of ocular hypertension, there was a significant decrease in spatial frequency in both the sham and the overrectomized animals. But what's really important to point out is that there was a significant worse spatial frequency in the overrectomized animals than the sham animals, indicating they had exacerbated injury or more impairments of visual function, which is actually hinted at before, that women may be subjected to worse visual impairments than males. This might be one of the reasons why. So this is repeated in the IOP independent injury model. So we looked at optic nerve crush again. Here, orange represents the sham animals, purple represents the overrectomized animals. Again, dashed lines are the control eyes. Again, there's no change from baseline through 12 weeks after injury. And again, what we see is that 4, 8, and 12 weeks after injury, both the sham and the overrectomized animals had significantly worse spatial frequency than their contralateral eyes, which is good. And again, by 12 weeks, the overrectomized animals had worse spatial frequency than the sham animals, again, indicating that they had worse visual function compared to the sham animals. So I think there's a lot of data here, right? And I went through it pretty fast, but I don't think I needed to go through a lot of the initial things. So in summary, again, I think this is very relevant to glaucoma. And I think a lot of basic science has shown that surgical menopause and estrogen has an influence on multiple factors related to ocular health and vision. So again, it affects intraocular pressure, outflow resistance, ocular mechanics, and gene expression. But it also obviously affects retinal ganglion cell survival, which I didn't talk about here. It affects inflammatory mediators, so actually on the protein level as well. And in injury, it affects visual acuity, so how well they can actually see. So all of these things play a role in health, in eye health, glaucoma, and progression. So I think it's a very important subject that we need to look into. And obviously, this is not something I could have done alone. So I'd like to thank all of my collaborators, former members of the lab, the current members in the lab, the financial support that has led to this work being completed. And I think that will be all for me today. So you are now free, I think, after our panel to enjoy cocktails. So let's get to it.
Thank you so much to all of our speakers for their excellent presentations. I want to open up the room to questions.
The clinical studies you showed, if you look at the odds ratios, they are statistically significant as the samples are large, but they are actually quite small. And if you think in terms of prognostic value, so an odds ratio has a relatively simple interpretation in terms of sensitivity specificity. So it would be like true positive over 1 minus true positive over false positive over 1 minus false positive. That's the. So what I'm trying to say is if you convert that to sort of like sensitivity specificity, let's say you need like big odds ratios to make a difference in terms of thinking about a prognostic factor for an individual patient. For example, if you have, let's say, 90% specificity and 80% sensitivity, you can reach, looking at the formula I just gave, that the odds ratio will be like 36, okay? And you're talking about like 1.1, 1.4. So my point is, what do you think, is it really something then that will make a difference in terms of having a prognostic value when you're dealing with an individual patient, because it seems to be so small, you know? So
Yeah, that's certainly a great point, and thank you for pointing that. For the odd ratios for the two population-based studies is 2.6 times, so 2.6 times that women enter menopause before age of 45 have increased odds of having glaucoma. And for the cohort study, for the oophorectomy study, that even it's 1.6 times. So 60%, I guess.
yeah, but it's 60% of something that is very small.
Right.
So that's why it doesn't really.
True.
My point is, does it really matter?
And for the for the African Americans, risk reduction that we found is about 50%. So it's like you can cut the risk in half. I know when we talk about like old study, right? Like you cut risk by half, and also that risk is also small. So it's probably similar. We look into the effect of it. What's the percent that we cut, or what's the proportion that we cut? But I understand what your point, that the risk is small, similar to the ocular hypertension too.
Thank you all. This is very informative, but this question is for all three. Maybe I missed this. After oophorectomy and menopause, estrogen levels will go down to a level that is similar to males, maybe a little bit lower. But I didn't see too many comparisons between men and women in these models, or in the animal models, comparing the oophorized to the males. If the effect is so bad that they became like males, then why do the clinical studies don't show a difference in the younger age between men and women? I mean, that's a question that's going to be applicable to all three talks.
So actually, I think this is a question many people discuss this. I think, why, the female, they have this reduced hormone similar to men, why they show the thing. I think the baseline difference between male and female before the premenopause are totally different. Their tissues are used to the environment regulated by the estrogen and the pathway. So in this way, suddenly when the estrogen being pulled away, so they have more significant impact versus male, you have the androgen and other things there to maintain. So in that way, the estrogen play a much less role. So I think that's my guess. But right now there are 4 factor models. You have the XXY, there's some animal model called the 4-core genotype. You could have, male carries 2X and Y. So in that way, you can tease out either it's a chromosome or it is due to the sexual difference could be due to sexual chromosome difference or the hormone difference. So this is really kind of one way to do in the animal models to tease out what's the difference. So, yeah.
Yeah, I can make a quick comment about that as well. In some ways, we have done males and females actually in a recent group. We presented at ARVO this year, where we do the optic nerve crush injury, male, female, and they actually have the same degree of injury. And again, the overrectomized females were worse than the shams, but also worse than the males. So the male animals didn't seem to be different than the sham animals, if that makes sense in the visual acuity, but the overrectomized animals were still worse than both of those groups. So it kind of hints at there might be something about the hormonal difference and not the chromosome, maybe not the chromosomal difference. Well, that's a study that has to be done still, but they didn't have exacerbated function compared to like the females after overectomy.
I have a mechanistic question, Andrew, related to what you presented and maybe to the entire team. So you assume that the mechanisms is related to the biomechanic model. I mean, you bring it back to that. But if I recall right, surgically induced menopause also causes a very profound peripheral vasoconstriction and elevated blood pressure. Is it possible that the mechanism is really second? And we've heard, I mean, estrogen also acts as a vasodilator. So we heard that the intraocular pressure is not that, I mean, we're talking about 1 to 2 millimeters. Is it possible that these benefits and effects are mechanistically actually through the vascular system?
In terms of I view kind of, it's a very complex disease, right? There's probably not one thing, this cardiovascular component, a biomechanical component. So I kind of view it as very complex in that sense, but I do think what you're hinting at is correct, where there is an effect on the cardiovascular system, especially with treatment, that is kind of hard to tease out which one is causing it. So I don't think it's a naive, it's an empty answer in a way, so I apologize for that. But I do think it is related to the cardiovascular changes, but it doesn't necessarily negate the impact that it has on the other parts of the system as well that need to be, that we need to pay attention to.
Yeah, I agree. I have 2 comments related to your questions. I think surgical menopause also have a very abrupt decrease in hormones. So it's totally like a new balance that the woman has to kind of go through, almost like an acute injury. But then the natural menopause perhaps have a little bit more transition into it. So usually women go into menopause transition between 45 to 55. So that time the body is starting to get used to the new hormones imbalance. So that one. The 2, I agree with you. I don't think that is only mechanical theory that will support this. It's not only IOP related. I think it's also vascular, because estrogen's receptors also present in the blood vessels. And this is probably gonna also impact and compromise, how the body responds.
Great session. This is sort of a follow-up to Alain's point. There's some thought that, or evidence that the translaminar pressure is important in glaucoma. And my question is, how do sex hormones and estrogen affect intracranial pressure? Have people looked at that? relative to intraocular pressure?
Yeah, that's a great question. I actually do not have that information, but I think that will be something that should be looked, into as well. And there's a lot of study in the aging of the brain as well, and how estrogen actually could protect Alzheimer's, for instance. So these could be kind of, because estrogen affect, just like you said, all the organs in the body. So I would assume that it probably have some relationship and related to that as well.
So I want to add one point, things this estrogen can really regulate the stiffness of the lamina curvosa. So maybe still the same or tiny change with the, the pressure difference. But if the lamina curvosa become more fragile, I think the impact could be more destructive, so.
Yeah, so, sorry, I just want to address that comment, and if you don't mind, 1 second, sorry. So it's actually interesting, and I'll keep this story short. The reason I actually started thinking about this and I started looking up papers on glaucoma and estrogen is actually because of IIH, idiopathic intracranial hypertension, which actually predominantly affects women, predominantly affects obese women, right, which is actually related to increased estrogen signaling.
We call it the fours.
The
Fat-
force.
Fat 40s female.
I never heard that term before, so I probably won't use that in a presentation, but that's interesting to know.
That's in medical school, dog.
Yeah, so there we go, different trainings. But I think the point is it probably does play a role, because when you think about that transition, most of those patients are female obese. And so we're trying to interested in, does it affect the biomechanical properties of the optic nerve sheath? Is that how you start having these changes in pressures? We don't know yet, but I think it's something we're kind of interested in looking into.
Also,
So I wanted to make a comment. So there was some comment that, you can't hear me? Okay. There was some comment that, you could look at males, right, as the control. But that's not entirely true. I've had some patients with gynecomastia and painful breasts in males. And if you're on spironolactone or those antihypertensive medications that affect the mineralocorticosteroid pathway, they have gynecomastia and breast pain because their estrogen levels are affected. So these are variables you have to pop out.
Yeah,
I want to go back. Sorry, go ahead.
So just I want to comment, there are actually crosstalks between glucocorticoid receptor and also mineralocorticoid receptors with estrogen receptors. So it's really kind of intertwined together. So I think,
That could totally confound your data. You're using males, and it turns out they're all hypertensives on a spironolactone, and they get no effect.
Wanted to go back to the point that Josh brought up. I'm not sure about. I know that in traumatic brain injury, estrogen was found to be useful, and there are situations where the intracranial pressure is higher there, and I think that they've shown that it can lower the ICP in that condition, but I'm not sure if that would be the same, for a person who doesn't have any brain injury.
So this was a great session, and I want to point out the excellent correlations between the low effect size, the effects on IOP in the randomized clinical trials, and the effects on IOP in the animal models. They're all the same. There's something there, and when we submitted the paper on the aromatase knockout mice, and it was like a 1 millimeter increase in IOP, the reviewers hated it. It was like, what is 1 millimeter of mercury going to mean? And how are we going to translate all of this to women with glaucoma? And I would say that more work needs to be done to find that second hit. It's like retinoblastoma, right? You have a 2-hit hypothesis. So you've shown that, the mouse or the rat that has ovarectomy, if you increase their IOP, they're even more sensitive to damage. But we need to distill that down further. Is there a gene or some other receptor that if you hit that, then you see a big effect? And I think people will start paying attention then.
Yeah. So in response to that, actually, when we cross the GPR1 knockout mice in the heterozygous with a 2 millimeter mercury increase, and then with NOS3 knockout heterozygous, most people think there is no phenotype with IOP. So we see also 2 millimeter mercury increase. When we cross the two together, double heterozygous, their IOP goes 9 millimeters, or almost doubles the IOP. So compared to the wild type. And so that's really kind of shows the interaction. And so another part is in collaboration with Anthony from last year. He did a look at the UK Biobank, the whole exome sequencing data. You probably forgot from the whole exome sequencing data with 40,000 individuals in the phase 1. So we did find GPR1, the G protein coupled estrogen receptor 1. So there are some really rare variants, about 2 and 3 variants. And so it's a borderline significant, it's a 10 to the minus 4th on the p-value. But the IOP change is 6 millimeter mercury on those individuals. So that's really kind of with the rare variants on GPR1 function. So probably have more bigger, IOP impact on the individual. So.
Yeah, I do agree. It kind of comments to your comment kind of points to the fact that these are almost, you'd think, subtle changes, right? Individually. But I view that as more of kind of like laying the foundation, right? So if everything's changing, gene expression's changing, IOP slightly changing, the second you get a second hit, your response to that second hit is going to be very different possibly than if you didn't have these changes. And that's really important. And I think to me, like I always use a parallel. I was kind of hoping, like, the talk to this morning that was supposed to happen with the cardiovascular parallels, right? So I always bring it back to cardiovascular field. And the reason I do that is early menopause is associated with a 1 to 2 fold increased risk of cardiovascular disease. Late menopause is associated with like a 0.8 reduction in cardiovascular disease for women. That's almost identical to what you see in the glaucoma case. The percentage of female and males in cardiovascular disease is almost identical to the percentage of females and males in glaucoma. The difference is menopause is considered a sex-specific risk factor independent of age for cardiovascular disease, whereas for glaucoma, we're still not quite there yet. And I think these studies, this epidemiological work, the genetic work, basic science work is kind of hoping, pointing in that direction that this is something that is fundamentally changing what's happening for women, at least. Sorry, go ahead.
Yeah, so no, I think it's all quite convincing. And for me, we often use epidemiology just to identify some novel biology, which we then might be able to alter in a different way with a more clinically substantial effect size. And, I think some of the stuff Yutao presented looked like, impressive effect sizes. But if I got you correct, sorry, my molecular biology is terrible. Yutao, you're saying it's like it's not obvious what you might put in a drop or you might inject in an eye that's going to work because it's all so complicated, yet you can see the effects by gene. So does that mean that a treatment, a gene editing or a gene therapy treatment might be the way forward? And the second question I wanted to ask, I just wanted to clarify this because I felt from the epidemiological evidence, it's not so strong for estrogen plus progesterone, it's more for estrogen only. Yet then in the other talks, I wasn't getting that. What's the final conclusion on that and what should we tell our patients? As in, if we've got, say, a patient at high risk of glaucoma or who's got glaucoma, who's going through menopause, should we be potentially saying if there are no other contraindications, go on estrogen only, or can we not say anything like that? Like what's the take home for that?
Sure. That's a tough one too. But because it also depends on if women have uterus or not. If they don't have uterus, so the choice is limited that way too. There's a newer maybe medication that it can take instead of progestin, coupled with estrogen. But we don't have data in terms of those women whether or not it's have the same effect as estrogen alone. So some might, then this is actually very consistent with other field as well in like other field in the WHI, when they look at the health outcomes, only estrogen alone that actually more protective than estrogen plus progestin. And that's just the one that we have data on.
So the common situation, which is a woman with a uterus going through menopause, in your glaucoma clinic, would you advise to go on an estrogen-only HRT?
Like I mentioned, because if they still have uterus, you don't have that option, right? So you have to have
right,
because if you have unopposed estrogen, you increase risk of women to have the endometrial cancer. That's why you couple that with progesterone. So it's not going to be just completely, we're not going to be able to generalize to women who have uterus, but there's a newer medication that you can couple with estrogen and instead of progestin, but we do not have data whether or not that actually have the same effect as the estrogen alone.
Got it, thank you.
Yeah, so for the therapy part, I think really right now is people use more the selective SERA, selective estrogen receptor agonist. And so it really depends on the receptor, that those alpha, beta, 5 different isoform, their relative ratio, because in one, the same chemical treatment, in some patients, if their mixture with different isoforms of the same receptor, you could have more dominant, antagonist reaction compared to someone with, the main isoform of the receptor. So that is an agonist. So it's really, we don't know how these different isoforms are regulated in different tissues. Yes. So like for cancer therapy, you could have the estrogen, one isoform, same thing. It's agonist for one tissue or antagonist for another tissue. Yes, there's a tissue specific people now actually taking regularly for, relieve some certain, menopause, symptoms there. So I think it's promising, but I think it's still, there's probably more kind of receptor specific, like a G1, G36 there for kind of either block certain receptor or the other ways. And, so because estradiol is really too general, it binds to, all kinds of glucocorticoid receptors, yes, and binds to ESR1, ESR2, GPR1, and their response, has instant response and then delayed response. And so they actually cause different things. And we try to treat the cells with, kind of conjugated estradiol. So in that way, it doesn't go to the membrane, go inside the cell. So we see different response there. So I think maybe that's clinical way, maybe control the isoform and the delivery to the tissue. So I think it's still promising, at least, we have so many libraries to screen and so many things to do. I think, I think it's promising, at least opens a new door. So.
We just have one question from the online audience. They asked, so testosterone also decreases after menopause. Could you hypothesize that it could affect IOP and risk for glaucoma?
Testosterone after menopause?
Yeah.
I'm not sure about testosterone after menopause. I think probably with the balance between testosterone and estrogen, probably different after menopause, maybe more closer to men than before. So what was the second part?
Could you hypothesize that it could affect the intraocular pressures?
It's possible as well, but we also have the evidence from the outflow, right, that's strongly supporting the lack of estrogen actually change the structure in the outflow. That could be the cause. Yeah.
There was a retrospective study, I think, that looked at testosterone levels in postmenopausal women and found it was associated with intraocular pressure.
And also from the neighbor study a long time ago, Jessica did the pathway analysis using the IOP. We did see the androgen-related signaling pathway also related to the glaucoma risk.
It's in men.
Yes, in men.
But this is women, yeah.
I just have a question too about, as we're talking about how this would modify counseling or treatment, and maybe not on an individual level, but on like a population level. So I thought the data around the effects in African-American women was very interesting. And that is also a population known to have much higher rates of hysterectomy and oophorectomy compared to white women, for example. So, and I think someone mentioned the difference between surgical menopause versus kind of a more gradual change that you might have in the absence of surgical menopause. And so I was just curious on what your thoughts on should there be, public health messaging around glaucoma risk with early, surgical menopause, because I don't think that's something that's well known in that community, and maybe that would kind of at least influence that decision making, because right now the rates of hysterectomy and oophorectomy are quite high in that population.
Certainly, I am a little biased, so I think that should be because of, there's a lot of evidence supporting that increased risk, right? Like Felipe said that maybe it's small, but it's actually there. In some women, maybe higher even. And we found that in the WHI, the effect is larger in African American when we give the estrogen alone. And we look into that and try to find why that might be the case. The first explanation could be just the effect size, like maybe the risk is higher and then we lower that risk compared to Caucasian with the risk is low. So you need a lot higher sample size to be able to prove that. And that's such as a claim database that Josh group actually showed that estrogen protective. But they didn't do the ratio analysis. But in our group, we didn't have the big enough white women to show that effect. Or maybe if we have the longer follow-up, we might be able to see that effect. So that's one. Second one is related to, like you say, women, African American women enter menopause earlier than the 6 to 12 months earlier than Caucasian. That could be, cut down the hormones that could have protected them, right? That's one. And also but we look at that, we didn't see in the statistical model to prove that. The third thing will be maybe there's some second hit that we were talking about, maybe is actually moderated through some kind of, like the pathway such as a nitric oxide synthase pathway, which may be more dysfunction in African Americans, and maybe they have two hit when they lost that protection early on, and they also have kind of borderline dysfunction of the nitric oxide synthase pathway. That might also, you see the larger effect when you give it back that protection.
So I have a little comment on that. Also, this morning, we have two geneticists that mentioned about family history. Yes, this is something accessible through your clinic. So if you find there are some family members have history of glaucoma, I think this is probably some women need to really follow up more closely. I hope the PRS score is more available for regular patients, but if they are in the top 10, 25%, I think this is also someone maybe need to follow up with the annual exam probably.
Yeah, that will be a question to you too, right? At what type of, like if we go back to look at the genetic risk scores and just redo the analysis, what would you pick to, to kind of see if add that and that would modify the risk?
I know Lou and I just chatted and said we're going to ask someone to look at whether there's a gene-environment interaction for age at menopause, and because if it doesn't matter what age someone's menopause is, if they're at such low genetic risk of getting glaucoma, they're probably hardly likely going to get it anyway. And so maybe in those who are genetically predisposed, the effect estimate would even impress Felipe. I don't know. Let's see.
No, in terms of giving hormone therapies, right? Like if you give hormone therapies and see if that modify the risk, what we found, like would genetic risk score of some sort would help to also kind of pick out who would respond.
Potentially.
We have one more. Oh, sorry. We have one more online question, or a question from the online audience. Any comments regarding hormone replacement therapy and gender transitioning, both from female to male and male to female, and the overall changes in these hormones with regards to glaucoma risk?
That's actually going to be another interesting study to do, right? I don't think we have a lot of information on that. No. Yeah.
A lot of it also is numbers, right? You need to be in a clinic where you have a substantial numbers to answer those types of questions, but it almost offers an ideal model in terms of understanding how hormones are changing would affect either, you could look at outflow facility, you could look at OCT changes, or visual acuity, just to know if those actually are affected as well.
So actually NIH has special RFAs for those proposals involve LGBTQ. So they are try to find any studies that people can submit.
Any comments from the panel about the fact that we're discussing potential treatment with a lot of work ahead of us for something that has been shown to have some carcinogenic effects?
You're referring to estrogen alone, you mean? Do you want to comment on the WHI study?
I mean, I'm just, just being a bit provocative here to.
I was going to let the
we're
clinician
talking
answer
about
first.
the treatment, but, we do need to understand that there's been some reports that have in the OBGYNs are concerned, at least some of them.
So your question was, sorry, can you repeat it? Is it?
Yeah, I mean, treatment has been associated with cancer, breast cancer.
Yeah, I think in theory, if you have the anti-estrogen and you disrupt that balance, I would assume, and I think the study would show. Do you know any other study?
I mean, that probably comes also back to the WHI initiative, right? But again, I was thinking you'd talk a little bit about the kind of secondary analysis they did when they comment not only on the type of estrogen given, but the timing of it and things like that. So those all actually play a role in how bad it is, or potentially how bad it is for females. But I also think it goes into what Yutao was talking about before, where it's a very complex signaling. It's not very simple, like what might be good for one tissue might be negative for another.
Exactly.
So it's very important, I think, ideally you'd have targeted delivery of anything related to this. It's tough. I view it as challenging when you're looking at hormones. Hormones are by design supposed to be affecting every tissue in the body, right? That's exactly how they're distributed. That's exactly almost every tissue on your body has receptor for hormones because it's the most efficient way to do those things. So it's very hard to have selective targeting of those hormones. And obviously in different tissues, they do different things. So from my perspective, first line is understanding the changes with this, identifying targets that we might be able to target that might be either independent of estrogen specifically, or finding ways to deliver it targeted where we don't have these systemic effects to reduce any of these complications.
Great.
Would you mind passing the mic up just one thing?
Have you considered the possibility that this effect is through the immune system?
I'm sorry.
Have you considered the possibility that the effects are through the immune system?
I guess, like, there are effects, like menopause has effects on the immune response. So again, that's why I put up the diagram of the eye. This thing affects, like menopause, estrogens, again, it's a hormone, right? So it's affecting things throughout the whole body, and it's designed to affect things throughout the whole body. So I think it's a very complex web of our understanding, and you can see from your studies with the genetic background, it's not just targeting one thing. You're going to affect a lot of things, and people have shown that it affects things in the immune response. It affects things in the inflammatory response and the innate immune response. So this is a very, very complex system, like a very complex, not system, but a very complex disease with a complex, menopause is a complex change. It's not just affecting one target, there's many targets being affected. So I think yes is the answer to that question, but to what level? I don't know, it might be even across the board, blood pressure, mechanics, inflammatory, immune response. But the fact that estrogen menopause is hitting on every single one of those levels, I think is what gives it, which should warrant attention.
Yeah, another way maybe to look at the repurposing of the current drugs available. We can look at how the current drugs also affects the estrogen signaling pathway to see if we can look at those. I think mainly is identify the individual patients who are related or affected by these pathways. So I think this is also, we can attach this to neuroprotection, also mentioned in the retina part. I think that's really, I think estrogen is not simple to the retina neuroprotection, probably affects many cells there, different cell type photoreceptor. So I think, anti, TGF beta effect. At the same time, we also need TGF beta fibrosis there to maintain our normal function. Yes, so it's really how do we maintain the balance, I think.
Okay, any more questions? No. So I think we have a break from now until 6 o'clock. I was told at 6 o'clock we'll be resuming with cocktail hour and then the dinner. So thank you for a wonderful session. Thank you.